For this training, there's a series of questions. You can pause the video right now and attempt at responding to them as a true or false statements. In this video, we're going to review the guidelines from EMA and FDA that refer to immunogenicity safety assessments. There are several guidelines that refer to clinical immunogenicity safety assessments. If you see, 4 of them have immunogenicity in the title. I did find others that refer to the need to follow immunogenicity safety assessment. If you look at the indentation, you can see that I put some of them to the left, like number 1 and 3, and the others are to the right because the ones that are to the right refer to either 1 or 3. So regardless of the title of the guideline, when you are looking at the safety assessments, they were very smart in just keeping all of the key elements in either the EMA guideline on immunogenicity assessment of therapeutic proteins or the FDA guidance for industry for immunogenicity assessment of therapeutic protein products. So they have the key elements that we need to look at. In addition, the gene therapy EMA and the oligonucleotide FDA have not only referred to the guidelines that I call number 1 and 3, but they also have additional safety assessments that we need to consider. So we're going to be going over those 4. Number 1, number 3, number 5, and number 6. In addition, there's the labeling from FDA that tells us where to put in the label and how to summarize these findings from clinical trials. Let's take a look at the definition of immunogenicity. By the FDA is the propensity of the therapeutic protein product to generate immune responses to itself and related proteins. The typical example would be an anti-drug antibody. This is generated by the drug and it's an antibody to the drug. But this is the second piece is very important, or to induce immunologically related adverse clinical events. In this case, these immunologically related adverse events are not directed or may not be directed to the drug. They could be directed to tissue. They could be different kinds of immune-related adverse events that we need to identify as having been triggered by the drug. The EMA guideline adds the important word of saying the drug is capable of inducing unwanted immune responses. It's shorter, but it basically states the same. Any kind of immune response that is unwanted and it's induced by the drug is considered an immunogenically related adverse event. So how do both guidelines guide us into what to follow in terms of safety? So in the consequences of safety, they are very clear that we have to consider 5 different potential adverse events that could be immunogenic, and we will go over those later in this video. At the end, the important thing is that both guidelines want us to do this assessment because we need to create a risk mitigation or a risk management plan that is based on the clinical phase. What did we see? What did we identify as potentially immunogenic? And also we need to identify what were the clinical consequences to create either mitigation or a management plan for the drug. The guideline from EMA on gene therapy and FDA on oligonucleotide development, both of them refer to additional immunogenicity assessments that are inherent to these modalities and may not be found in other protein therapeutics. So on the left, I listed the ones from EMA, and on the right, the ones from FDA. Both of them are saying that we need to consider the whole drug, but also we need to consider the components like the vector or the delivery system or the carrier, depending how they call it. And most importantly, these two are using gene therapy, and the immunogenicity created by the nucleic acid sequences or the oligonucleotide that's in the drug are important. The EMA also talks about immunogenicity to the transgene product. So all of this has to be evaluated. Are any of these components or the whole drug capable of mounting an immune response that is unwanted? Now, the EMA talks about the product of the transgene because this is under the section that's called unwanted consequences of immunogenicity. So if you have an immunogenicity to the product of the transgene is not within the drug, this product will be created by the person who received the drug, but it's still immunogenicity to the treatment. So that has to be considered. In addition, the FDA pays a lot of attention to potential immunogenic capability of backbone modifications. And in their draft guidance of June 2022, they're already thinking about nanoparticles and how the different mRNAs in recent lipid nanoparticle vaccines have a potential of creating more or less immunogenic responses. And they're talking about modifications to the RNA. So RNA that's modified, or for instance, the self-amplifying RNA, etc. So they're open to future advances in medicine. So that's why they're talking about backbone modifications, because they're talking in this section about unwanted immune response to an oligonucleotide, either the messenger RNA for the product we need or modified RNAs. They do not talk in this section about potential immunogenic responses to the product because this section is the section on oligonucleotide immunogenicity. Later in the same guideline, they do talk about also potential immunogenic responses to the drug. At this point, let's take a look at the first few questions. The one is there's two main guidelines for clinical immunogenicity safety assessments, one from FDA and one from EMA, and this is true. The goal is to identify potential unwanted immunologically related AEs that are likely due to the drug, and this is also true. These two guidelines further specify. The gene therapy guideline for EMA says you need to look at potential innate system responses that are characterized by cytokine elevations and inflammation. But you also need to look at adaptive immune system response. It could be antibodies, it could be cytotoxic lymphocytes, or cytokine secreting T lymphocytes. And it could be either to the vector or to the transgene product. And as we saw, they also say to the whole drug. So basically, they're telling us you need to consider absolutely everything from the moment you're starting your development program before it goes to clinical, before it goes into animal models. You need to see if there's any trigger to innate or adaptive responses so that if it goes to the clinic, we know in the clinic what do we need to follow up in these patients. The FDA is telling us in a different way that we need to identify potential immune responses that are triggered by the product. And this is where they're talking about, the sequence of that product, that oligonucleotide. They also tell us the pharmacology of the product. So the mechanism of action could actually increase the T cell response, for instance. And remember that we're saying unwanted immune responses. So in this case, if we have a pharmacology that is increasing T cell response like checkpoint inhibitors, what would be unwanted would be the degree. If this potential T cell response is more than we want and it's producing a safety concern, then it would be immunogenic, even though it's pharmacology. And this is where the expression profile also comes into this particular section. It could be not necessarily the oligonucleotide, but now we're talking about the pharmacology has to do with the product that is the result of that nucleotide transcription. And they also say you need to look at patient characteristics. Is there anything in this particular patient population that would increase a potential unwanted immune response? How is the immune system in these patients? And this is important because through the development, we have different patients. We may have actually healthy volunteers in the clinical trial. And of course, at the end, we're not pretending to have a drug for healthy volunteers unless it's a vaccine. But as we go along, the patient population might change. Also, if new indications are added, a new risk assessment has to be updated for the particular patient characteristics in terms of potential risks for immunogenic responses. So I generated this table, and the disclaimer is this is my interpretation of the guidelines. So you may have a different interpretation. But what I did was to list on the first column the different assays that are mentioned throughout the guidelines. And each one of the columns to the right, the header, refers to what is the component of the product that is being recommended for which assay. Then what I did was to say a green checkpoint is for expected, which is listed more clearly in the guideline. In the red is the ones that the guideline says exclude these patients from treatment if they have this particular assay that's positive. But then I put a blue one, which is evaluate, and that's a tricky one because there's a lot of different mentions of potentially saying, well, it would be recommended or it would be a good idea. So let me walk you through the table. First thing is anti-drug antibodies to the vector, and T-cell assays to the vector are very commonly done and recommended in the guidelines for gene therapy. Anti-drug antibodies to the gene product, if they're neutralizing or not, and T-cell assay are also very clearly mentioned. They would be triggered if there is an adverse event that happens after the gene therapy has happened. But these are things that would need to be followed depending on the clinical manifestations. Now, in terms of the neutralizing antibodies to the vector, these are generally exclusionary because if the vector cannot reach the particular organ, the vectors are used based on their tissue tropism, then it would not be able to inject, let's say, the gene therapy in the appropriate tissue. And if the vector cannot deliver the gene, then the therapy doesn't work. So in general, for gene therapy, the neutralizing antibodies to the vector are tested with assays that have been also approved, or they are a companion diagnostic with the therapy, and it's exclusionary. Now, the rest of it is basically mentioned in the guidelines. So you have to test different things for the whole drug. For the vector, it would be good to also see if there's a T-cell response or a complement activation to the vector. This could happen oftentimes with liver tropism. But also to the gene product, especially, for instance, in Duchenne's muscular dystrophy, is there a problem with muscle reactions to the gene product based on either complement activation or T-cell response. So basically, they are mentioned and they could be triggered based on clinical manifestations. Let's add to these assessments my interpretation of what the FDA guidance on oligonucleotide therapeutics is telling us. They do pretty much agree with or align with what I interpreted from the EMA guidelines. Now, they also mentioned some things to consider, so I added another checkpoint, which is the yellow one. So they do say to assess or consider anti-drug antibodies and T-cell assays to the oligonucleotide also. But consider a potential innate cytokine to the delivery strategy. This particular guidance from draft guidance from 2022 is telling us a lot about saying that lipid nanoparticles are starting to be used a lot for mRNA products, and maybe there's something in the lipid nanoparticles, or for antibody-drug conjugates, you have a linker. So all of these potential elements of your drug that are used could be immunogenic, and they could trigger either innate or adaptive responses. In terms of the oligonucleotide, they do recommend to assess innate and adaptive responses and complement activation secondary to a response to the oligonucleotide. And remember, they say the backbone modifications to it. And also they do say that, we need to check innate cytokine to the gene product. So my interpretation from both guidelines is that it is not as simple as it may look like. It's not just one, like for monoclonal antibodies that are not complex biologics. You may have just one assay for antidrug antibodies and one assay for neutralizing antibodies to the drug. But in these cases where we're getting more complex products and they are combinations of different elements, it's becoming a bit more complicated on what part of the drug is immunogenic and how do we assess it. Okay, time for the other questions. The risk assessment focuses on identifying innate and adaptive response considering the product and the pharmacology. What's missing here is the patient. That is an important thing that's really mentioned. Both guidelines talk about innate and adaptive responses, but remember the patient considerations are very important for the risk assessment. So don't forget that. That's why this is false. Antibodies to the drug product with neutralizing activity are always exclusionary to receiving the drug. This is basically for the vector. They don't mention this for drug product, so this is the reason why this is false. Thank you very much for watching this video. I hope you found it informative, and I hope that you learned something from watching it. Please remember that I have other videos in my channel, also educational videos, and consider subscribing to my channel. Thank you very much and have a good day.